Shall we start, ma'am? Sir, shall we start, sir?
Yes, go ahead.
Okay. So, good evening to one and all. It's indeed our honor to host another session of Advances in ART Masterclass series today. Today, with a unique twist that we have original research being presented from Raju sir's team from Krishna IVF. So, we have Raju sir, Dr. Kavitha ma'am, and Dr. Kavitha Lakshmi ma'am. So, before we go into the details of today's section, we'd just like to reinforce that SCALE has started in association with TAPISAR. We have started 2 fellowship certificate courses, one genetics and infertility, which is for 12 weeks, and immunology, reproductive immunology course, which is for 8 weeks. The registrations are on and we plan to start in October end. With that, I'd request Vasundara ma'am to kindly introduce sir and Kavitha ma'am.
Warm welcome to all the delegates for yet another masterclass. This is my great honor to introduce my mentor and most inspiration. I've had lots of inspiration from Dr. Raju sir. Many students have trained across India with his wisdom and knowledge. We keep learning every day. So, sir is an emeritus professor at Human Genetics, Andhra Pradesh, and he's a honorary professor, adjunct professor, program director. He's got so many research publications and citations. So, if you just type his name, you will get a list of publications and research citations. Lot of credits. More so, I have learned a lot on day-to-day, philosophy, and sir carries a huge fan following, and he's such a great teacher that all of us want to learn from him. Thank you so much for accepting the invitation from the SCALE team and doing the follow-up of our part 1 FSH part 1 polymorphism series. This is going to be part 2. As Dr. Sindhuja said, this is going to be unique because can I have Dr. Kavitha Lakshmi and Dr. Kavitha sleep? So Dr. Kavitha Lakshmi, Dr. Kavitha is a PhD and also having an MD in gynecology. This is a unique combination. Doing research is not easy, and she has done her research in human genetics from Andhra University, Visakhapatnam, and she's currently working with the team of Krishna IVF. And Dr. B. Kavitha Lakshmi is again another MS in obstetric and gynecology with a PhD scholar in human genetics, again from Andhra University, Visakhapatnam, and she's currently working with Krishna IVF team as a senior consultant along with Dr. Raju sir. It's indeed a great privilege. The uniqueness of this program is that they have their FSH receptor polymorphism as their thesis, and we are going to learn from the masters who have done research in this field. In the part 1, we did learn about a lot of LH receptor polymorphisms. We now know that receptor polymorphism is very, very important because we need to understand there can be a threshold for activation of these receptors. Ovarian response to stimulation is also dependent on these receptor polymorphisms, and sir has told a lot about LH receptor polymorphism. It can be of various types, AA, AG, or GG, and based on that we know that which is going to have a normal sensitivity or partial resistance, or if your GG genotype is going to be more resistant. Sir gave us a lot of insights, and after that sir also shared a podcast with us to understand the physiology of receptor polymorphism. With this small introduction, I would just invite Dr. Kavitha Lakshmi and Dr. Kavitha to one by one please share your insights today and more on FSH receptor polymorphism. And at the end, sir will also give us all the summary and take-home points from the entire lecture. Welcome, all of you.
Thank you.
Thank you, Dr. Vasundara, for your kind introduction. Are my slides visible?
Not yet, ma'am. Yes, ma'am.
Are they?
Yes, ma'am. It's not on slideshow yet, ma'am.
Okay. Give me 1 second.
Sure, ma'am.
Are they visible now?
Yeah, visible.
ma'am.
Good evening, everyone. And thank you so much for taking time and joining this webinar.
Ma'am, there is a chat box that says captions and subtitles. Can you please close that now?
Yeah, sure. Just 1 second, give me 1 second.
Sure.
Good evening, everyone. And thank you so much for taking time and joining this webinar. And I thank Dr. Vasundara and Dr. Sindhuja for giving me this opportunity.
Slides are not visible, madam.
Sharing
Yeah, keep sharing.
Yes,
Yeah,
perfect.
visible.
Yes, ma'am.
Yeah, with So over the next 15 minutes, I'll be speaking about FSH, follicular stimulating hormone receptor polymorphisms, and its role in assisted reproductive technology. The outline of my talk is Introduction to Polymorphisms and Receptors, Follicular Stimulating Hormone Receptor and its action, Follicular Stimulating Hormone Receptor 680 and 307 polymorphisms, role of receptor polymorphisms in ART, results of retrospective study conducted at Krishna IVF Clinic, followed by conclusions. So, the word polymorphism in genetics means occurrence of two or more different forms of particular gene within a population. And these different forms of genes should appear at least in 1% of the population. And they usually arise due to naturally occurring DNA sequence variations at a specific site on a chromosome. And these polymorphisms, they usually lead to different versions of genes among individuals of population. And the most common example, this is the ABO blood groups in the population. And then polymorphisms can be as small as single nucleotide change called SNP, or they may involve larger stretches of DNA. Sorry, I think I have, yes. And now what are receptors? These are specialized protein molecules located on the surface of the cell or inside the cell. And these receptors recognize and bind to specific molecule called ligand, such as a hormone or neurotransmitters or drugs. And these receptors transduce signals that trigger a cellular response. And these receptors also enable regulation of various biological processes, including growth, metabolism, and immune response. I'm sorry, there is a... Okay. And these receptor, what are these receptor genes and polymorphisms? Now I have spoken about polymorphism. I have also spoken about receptors. And what are these receptor genes and polymorphisms? Receptor genes are nothing but DNA sequences that encode for protein receptors. And the polymorphisms in these receptor genes are existence of multiple genetic variants for the receptor genes. So most of these receptor polymorphisms are single nucleotide polymorphisms, that is called SNPs, and there is a change in amino acid sequence or expression levels of these receptor proteins. So most of these naturally occurring genetic variations in these receptor genes, they do not cause disease, but they may impact biological processes and clinical outcomes, especially in treatment scenarios. So they can alter the receptor sensitivity, ligand binding, intracellular signaling, and bring about differential clinical response to medications. So follicular stimulating hormone receptor is nothing but it's a receptor expressed on the granular cells, mediating the action of FSH on them, leading to ovarian follicular genesis. This follicular stimulating hormone binds to follicular stimulating hormone receptor, initiating the signal pathway, leading to regulating of the follicular growth and estrogen synthesis. This receptor activation, it stimulates aromatase activity, increases the estradiol production, which is essential for a dominant follicle development. So when you look at the action after the hormone binding to the receptor, there is activation of the G-alpha protein receptor at the level of the internal molecule of the cell, and then it stimulates the adenine cyclase and increases the intracellular cyclic AMP, and then this cyclic AMP in turn activates protein kinase A, leading to transcription factor activation in nucleus, and this in turn brings about gene transcription and protein synthesis, leading to follicular growth, estrogen production. So what happens in these follicular stimulating hormone receptor polymorphisms? This is a receptor polymorphism is a normal genetic variant, may not show up any alteration in normal situations and may go unnoticed. But when it comes to controlled ovarian hyperstimulation, this normal monofollicular development is completely disrupted, leading to polymorphisms, showing a different stimulation response to individual variations. And what happens in a stimulated cyclist? There is a modification in the receptor sensitivity to FSH, altered ligand binding, and it can impair or enhance signal transduction efficacy, leading to impaired or enhanced cellular response. So what is the role of these receptor polymorphisms in IVF? So as you all know, controlled ovarian hyperstimulation with gonadotropins is the crucial step in IVF treatment. So starting with the right dose of gonadotropins, this can lead to optimal follicle to set yield. But current dose duration is based only on phenotypic markers like age, BMI, antral follicle count, AMH, etc. But not all women respond equally to the same dose of FSH in IVF stimulation, often leading to either hypo or poor response or hyper response. Variability in this response suggested a genetic basis for the differential ovarian sensitivity to stimulation. So these first polymorphisms were identified back in 1995 by Atimoho. So he is the first person to identify genetic variants in FSHR gene. So later, this work is followed by Professor Smith and Green, Manuela Simoni, Ulaf, and et al. And these people, they expanded the detailed knowledge of FSHR receptor biology, crucial for this reproductive medicine and IVF applications. So FSHR 680 polymorphism is the most commonly occurring polymorphism in follicular stimulating hormone receptor, and it is also one of the most widely studied polymorphism in IVF setup. So this is a single nucleotide polymorphism in the FSHR gene. There is a substitution of serine with asparagine at the codon position 680 of the receptor. This particular polymorphism is located in the intracellular domain of the receptor. This is crucial for receptor activation and downstream signaling. So moving on to receptor 307 polymorphism. This is also a single nucleotide polymorphism. Substitution of threonine by alanine at codon 307 of the receptor gene in exon 10. So this receptor polymorphism is located at the hinge region of the receptor, connecting the hormone binding domain to the transmembrane domain. This receptor polymorphism exhibits linkage disequilibrium with the FSHR 680. These two combination of genetic variations occur together. So along with follicular stimulating hormone receptor 680 polymorphism, it is known to influence the FSHR response. LH-CGR polymorphism at 312 position. This is also a single nucleotide polymorphism. This has been extensively studied, LH-CGR polymorphism, located in the extracellular domain, specifically in the extracellular loop, which involves in receptor binding and ligand activation. Here, there is substitution of asparagine to serine at this position. Variation in this polymorphism may influence ovarian response and pregnancy outcomes. So our study, moving on to our study, that is a retrospective study on follicular stimulating hormone receptor 680 and LHCGR312 polymorphism and phenotypic parameters and their role in ART outcomes. In our study, we compared the dose. We allotted the alleles as G and A based on the amino acid present at that particular level. We have allotted G for when there is a serine amino acid serine, and then A for the asparagine. So based on this, people were categorized to GG when there is serine and serine, GA when there is serine and asparagine, and AA when there is asparagine. And in the similar way, for the polymorphism at LHCGR312, so AA was denoted for asparagine and asparagine, AG was denoted for asparagine and serine, GG for serine and serine. The distribution of these polymorphisms among our study, which includes 1,714 participants, the GG polymorphism included 18% and GA 47% and AA 33%. So the observations which we made in our study were dose of FSH needed across the genetic groups, number of days of stimulation, oocytes retrieved, number of mature oocytes, fertilization rates, embryos formed, clinical pregnancy rates, and live birth rates. So in our study, when we compared the dose required across the different groups, we observed GG variant required lower dose compared to GA and AA groups, but the difference was not statistically significant. But in case of women over 35 years of age, there was statistically significant difference noted across different genotypes, with GG requiring a lesser dose compared to AA groups. In a live birth rates, no much difference was noted across the genetic groups in younger women. But in women over 35 years of age, the rates are notably lower for GG, GA, and AA groups compared to GG, and the maximum live birth rates were seen in GG groups. So the inference from this observation is older women with AA genotype require the highest FSHR dosage, highlighting the combined effect of genetic variation and age-related changes. And we did not find any difference in atrial follicular count across the genotypic groups, no difference in number of oocytes obtained, no difference in mature oocytes, fertilization rates, or blastocyst formed across the different genotypes. And later, we did a combined analysis of follicular stimulating hormone 680 and LHCGR312 polymorphism groups. So we found individuals with the combination of follicular stimulating hormone 680 GG and LHCGR312 AA required a lower total dose of recombinant FSH compared to other groups like AA and NAG and AA and GG groups. And in case of clinical pregnancy rates, the follicular stimulating hormone 680 GG and LHCGR312 GG groups had highest clinical pregnancy rate up to 55%, whereas in case of follicular stimulating hormone 680 AA and LHCGR312 AA groups, the clinical pregnancy rates were lowest with 36%, and this has showed a statistical significance with a p-value of 0.02. Similarly, when it comes to live birth rate, the follicular stimulating hormone 680 GG and LHCGR312 GG, there was highest live birth rates with 38%, and in case of follicular stimulating hormone 680 GA and AA groups, the live birth rates were lowest with 24%. And there was a statistical significance with a p-value of 0.02. So to conclude, women with follicular stimulating hormone 680 AA genotype required a higher dose of follicular stimulating hormone compared to other groups, but older women more than 35 years of age with AA genotype required a highest follicular stimulating hormone dose, And then genotypic guided LH supplementation appears beneficial when considering combined FSHR and LH-CGR profile. Lower pregnancy and live birth rates in women with follicular stimulating hormone receptor 680AA and GA genotypes, though not significant, not statistically significant. And combined follicular stimulating hormone receptor and LH-CGR polymorphisms suggest higher success rates for certain genetic profiles like GG variants, and the pregnancy rates were lowest with AA genotypes. AFC and BMI independently predict ovarian response with limited interaction with follicular stimulating hormone receptor 680 genotypes. So age, AFC, and BMI to be considered first for dose decision. Genetic markers provide additional insights for personalized treatment strategies. So potential for this genetic profiling is follicular stimulating hormone 680 and LH-CGR 312 polymorphisms together may serve as biomarkers for tailoring controlled ovarian stimulation protocols. Combining genetic and phenotypic data could optimize this ART outcomes. Thank you.
Kavitha Lakshmi will start in.
Good evening, everyone. Today I am going to share our receptor work on the assessment of FSHR minus 29GA in combination with LH-CGR 18 insertion LQ and aspargine 312 serine gene polymorphisms as predictive genetic markers for ovarian response and reproductive outcomes in women undergoing in vitro fertilization with agonist protocols in assisted reproductive technology. FSHR minus 29GA polymorphism with the RS number 1394205. It is a single nucleotide polymorphism in the follicle stimulating hormone receptor in 5 untranslated promoter region. Substitution of adenine in place of guanine in minus 29 promoter region is associated with the changes in the receptor expression due to changes in the transcription factor binding sites. Mostly, the promoter region is present in the upstream of the gene, almost more than 10 nucleotides away from the transcription start codon. RNA polymerase, an enzyme which is useful for making the mRNA copy from the DNA in the nucleus, it enters to this promoter region and walks along the gene for a while till it reaches a transcription start codon and starts transcribing the mRNA copy and stops once it reaches the stop codon. This precursor mRNA gets modified into the mature mRNA and enters into the cytoplasm. When it is recognized by the ribosomes and FSHR protein synthesis takes place, and this receptor gets embedded in the cell membrane. And this promoter region is also a binding site for many transcription initiating factors. So any alterations in the promoter box results in the alterations in the rate of transcription. And they do not alter the sequence of nucleotides in the mRNA or amino acid in the protein. Experiments using the Chinese hamster ovary cells have demonstrated that A allele at minus 29 location, it leads to the lower transcriptional activity than the G allele. And Nakayama et al. In 2006 and Wunsch et al. 2005, they stated that this polymorphism at the minus 29 position occurs within a sequence important for binding a specific transcription factor, and presence of the A allele may disrupt this binding site. Unsheta 2005 showed no association between FSHR minus 29 GA and basal FSH levels or E2 concentrations in experiment done in 202 women. Ekreker et al. In 2009, they showed AA genotype association with decreased preantral follicle count and the number of oocytes retrieved and lower pregnancy rates in 50 Indian women. VIZAG et al. In 2011, on 100 subjects, they stated that 72% of the subjects with AA genotype at position minus 29 of FSHR gene were associated with reduced ovarian response and also conducted function studies on granulosa cells and found reduced mRNA expression and receptor protein expression in AA genotype. The magnitude of structural change value in the DNA topography of FSHR gene revealed that the DNA with the A allele is less accessible for binding of transcription factor compared with the G allele. In 2006, Dahlia Tohlab, Manchester, UK, they also found no relation with preantral follicle count and total dose and the number of oocytes retrieved. Due to the prevailing uncertainty regarding the utility of the FSHR promoter gene variant as a predictor of ovarian response and its outcome, this study aimed to examine its relationship with the ovarian reserve parameters, and the primary endpoint is the total gonadotropin dosage requirements in each genotype, and secondary endpoint encompassing the clinical pregnancy and live birth rates within a large cohort of 1,714 samples. Our study design was a retrospective cohort done in a single center, that is a Krishna IVF clinic, and 1,714 patients were taken and all patients were followed long GnRH agonist protocol, and recombinant FSH and recombinant LH drugs were used for the stimulation, and patients with a severe male infertility and oocyte donors and protocols other than the agonist and patients with a single ovary and significant medical conditions were excluded from the study. Uniform clinical and lab teams and the data from the FileMaker version 20 and data analysis was done with SPSS version 21. Coming to the variants, minus 29 GG, that is both alleles containing guanine, and GA, one with a guanine and other with adenine, and both alleles with the adenine is minus 29 AA. In total, 1,714 women, GG accounts for 44.6% and GA for 40.25% and AA 15.11%. Coming to the results, baseline characteristic across the FSHR minus 29 GA, small age difference was noted, and in this GA was slightly older than the GG group, and no significant differences in AFC and BMI were noted. Coming to the stimulation doses, no statistical significant difference was observed in the total recombinant follicle stimulating hormone dose with a p-value of 0.1. Coming to the oocyte yield, the GG genotype was associated with a statistically significant increase in the number of oocytes retrieved compared to the GA and AA genotypes. This effect, however, became non-significant after adjusting for potential confounding factors like female age, AFC, and BMI. Coming to the clinical pregnancy and live birth rates, a marginal association was observed between FSHR minus 29 genotype and clinical pregnancy rate. Individuals with the GG genotype had a slightly higher clinical pregnancy rates compared to those with the GA and AA genotypes, and this association became weaker after adjusting for potential confounding factors, and no significant association was found between FSHR minus 29 genotype and live birth rate. Chapter 2 assesses the combined effects of FSHR minus 29 GA with LH-CGR and 312S on ovarian response and IVF outcomes. LHCGR and 312S polymorphism, it is an SNP in the LHCGR gene located in the exon 10 on the extracellular domain, specifically in the extracellular loop 2, which is in the receptor binding and ligand activation site, and substitution of asparagine to serine at this position. And this polymorphism is close to the glycosylation site, so it could influence the receptor sensitivity variations in this polymorphism and influence ovarian response and pregnancy outcomes. Coming to the variants, 312AA is both the alleles containing asparagine, the AG is asparagine and serine, and 312GG is both containing serine. Coming to the results, stimulation doses when both combined, there was no statistical significance when FSHR minus 29 alone was taken with a p-value of 0.1, and some trends were observed with a p-value 0.07 for LHCGR and 312S, and no significant interaction effect was noted when both were combined with the p-value 0.14. Genotype combinations of FSHR minus 29GG and LHCGR minus 12AA, lower total dose of FSH was required. This was mostly 2,514 units. But for genotype combinations of FSHR minus 29AA and LHCGR minus 312GG, they need a higher total FSH requirement, a dose of about 2,722 units. And remaining genotype combinations, the total FSH dose requirements ranges from 2,750 to 2,865 international units. Coming to the oocyte yield, there was a statistical significance for FSHR minus 29 with the p-value of 0.04, and statistical significance also noted for LHCGR, the p-value of 0.003, but no interaction effect was noted when both were combined with the p-value of 0.34. Coming to the clinical pregnancy and live birth rate, FSHR minus 29 GA, it has a statistical significance with the p-value of 0.04, and for LHCGR and 312S, statistical significance was noted with the p-value of 0.01, and the interaction effect also showed a trend with the p-value of 0.07 when both were combined. Coming to the live birth rates, FSHR 29, no statistical significance noted, and LHCGR and 312S with the p-value of 0.03, statistical significance was noted, and no significant interaction effect was noted when both were combined with the p-value 0.20. But some trends were observed. The FSHR minus 29 GG and LHCGR 312AA combination, they showed a trend towards requiring a lower dose of medication and higher pregnancy rates compared to FSHR minus 29 AA and LHCGR 312 GG combination. All G alleles, FSHR minus 29 GG and LHCGR 312 GG in combination, they have a higher pregnancy rates when compared to FSHR minus 20 AA plus LHCGR 312 AA group. Chapter 3 assesses the combined effects of FSHR minus 29 GA with LHCGR 18 insertion LQ on ovarian response and IVF outcomes. LHCGR 18 insertion LQ, the luteinizing hormone and the human chorionic gonadotropin receptor, it was involved in the insertion of 6 base pairs, that is CTG-CAG at the nucleotide position 54 with the RS number 4539842, an insertion of 2 amino acids, leucine and glutamine in the signal peptide region. Structurally, this variant affects the LH changing and how the protein folds and moves within the cells and inserts into the cell membrane. In terms of the function, this showed increased activity in the cell structure, possibly because it moves more efficiently and has higher levels on the cell surface. Coming to the variants, 6 base pair insertion, both alleles with the presence of insertion, 1 insertion, 1 absence of insertion, and absence of insertion in the both alleles. Coming to the results, stimulation doses, There was no statistical significance when FSHR minus 29 alone was taken, the p-value of 0.1, and no statistical significance with the p-value 0.4 for LH seizure 18 insertion LQ, and no significant interaction effect was also noted when both were combined with the p-value of 0.39. Coming to the oocyte yield, there was a statistical significance when FSHR minus 29 alone with the p-value of 0.05, and no statistical significance in the 18 insertion LQ with the p-value 0.36. No significant interaction effect was also noted when both were combined with the p-value of 0.51. Coming to the clinical pregnancy rates and live birth rates, FSHR minus 29 GA showed a statistical significance with the p-value of 0.04 and no statistical significance in the 18 insertion LQ, and no significant interaction effect noted when both were combined. Coming to the live birth rate, no statistical significance in the FSHR minus 29 alone with the p-value 0.1 and no statistical significance in the LH seizure 18 insertion LQ alone with the p-value 0.9, and no significant interaction effect was noted when both were combined. But some special trends were observed. These are the combination of FSHR minus 29 AA and LH seizure exon 1 insertion LQ. They showed a trend towards a higher number of retrieved oocytes. Combination of FSHR minus 29 GG and LH seizure exon 1 insertion LQ, they exhibited a higher rates of clinical pregnancy and live birth rates. To conclude, the FSHR minus 29 GA variant shows minimal influence on the ovarian response and IVF success, indicating it is not a strong standalone predictor. Combinations of FSHR 29 minus 29 AG and LH seizure N312S and LH seizure 18 insertion LQ polymorphisms, they may modulate the ovarian response, but their overall effect remains modest. Patients' age, ovarian reserves, treatment protocols, and environmental influences, they significantly overshadow the genetic effects. Genetic testing for FSHR minus 29 GA alone offers a limited utility in guiding the IVF decisions, so a multifactorial approach is essential. Strengths of our study are, it is a large single-center cohort with 1,714 patients with uniform protocol and 2 independent combined analysis and detailed genotyping workflow and consistent lab conditions. Limitations are, it is a retrospective design and with the potential residual confounding and small number for insertion LQ homozygotes, and generalizability is limited to the Indian Asia Pacific population, and marginal p-values which needs cautious interpretation. Future directions were prospective multi-center validation with adequate power for insertion LQ homozygotes, and functional assay should be conducted on the granulosa cell FSHR and LH seizure expression versus the genotypes, and interaction with other gene variants and environmental factors, and integrate deep learning based follicle tracking with genotype guided dosing, and cost effectiveness of pharmacogenomic controlled ovarian stimulation in India. Thank you.
Thank you, Dr. Kavitha and Dr. Kavitha Lakshmi for your wonderful presentation. May I now request Rama Raju sir to share his insights, and then we can go into the Q&A.
As part of the Delphi consensus globally, which was conducted by the European group and the American group, where they studied the literature about FSHR receptors and LH receptors. The data about LH receptors is very, very clear. In the role of hyperresponder and in the role of normal responder. When it comes to FSHR receptors, AFC, age, BMI makes a major role for prediction of dosage. Take an example of oocyte freezing. when you have a particular FSHR genotype, you might need more number of oocyte freezing for a particular genotype because it is associated with a lower live birth per egg retrieval for a particular genotype. That is where this has a role. Similarly, your dosage fine-tuning will have a role. Unless you the first thing is when you're looking at FSHR receptor, the first and the foremost thing is you should standardize your dosage, and over on top of it, you can adjust your dosage. Unless you do not standardize your dosage, these receptors of FSHR will add to confusion and nothing at all. That's my carry-home message. Unlike LHCGR, where hyperresponder is associated with a particular genotype, the data about FSHR has many variables, and the combination of genotypes that is affecting it. If you are looking at a woman with an antral follicle count of 5 or 6 with a particular genotype, there it makes a whole lot of difference in getting an optimal dosage based on the AFC mesh, and there the quality of oocytes will also make a difference. For example, if you have looked at the data, FSHR A by A in 680 had a lower carry home baby rate. That means the number of oocytes that are needed to give a live birth might be different. There is a concept called how many oocytes will give how many babies. And the data is as follows: 100 oocytes will give seven and a half babies. That has not changed in the last 40 years. The equation is as follows: out of 100, not all are M2 oocytes. Not all M2 oocytes get fertilized. Not all fertilized oocytes go to blastocysts. Not all blastocysts get implanted. Not all implanted embryos will end in a live birth. So 100 oocytes will give 7.5 babies. This has not changed in the last 40 years. And on top of it, if you overlay the FSHR receptors, there it might make a difference. Similarly, on top of it, if you are doing a oocyte freezing, this receptor number that is needed to get a baby also changes. But at this moment, from our understanding, we are trying to look at the combinations, and we realize that there are 64 combinations that come when we add FSHR 680, FSHR 307, FSHR 29, LHCGR 6 base pair insertion, LHCGR 312. All of them has each 3 types of subdivisions. We are getting 64 data sets, and we are finding subgroup analysis which is showing interesting trends. So my carry home message is the concept for LHCGR is clear. The concept for FSHR is evolving, and I would not suggest routine FSHR testing for any patient unless you're doing in a research setting. And here in our clinic, we emphasize and we charge only for the LHCGR, even though the pricing includes everything. But the data that is used is only the LHCGR. And FSHCGR, it's still evolving, and we are trying to understand. And this PhD thesis has given us more understanding in the subgroup analysis. That I would like to say. And I would like to share some of the So if you look at this slide, there is no impact on the total FSH dosage. There's a weak association with the oocyte yield. And the most important thing is it should not be used as an isolated marker. Unlike LHCGR, you can use it as an isolated marker. The next area is still when you look at FSHR polymorphism, the genotype does not dominate. The phenotype dominates in the stimulation protocol. That means your age, AFC, and BMI is the main predictor of your dose, but not the FSHR phenotype. There are patient subgroups which might make a difference in future. When you combine with LHCGR, there is a link to oocyte yield, and there is a link to type of G allele or A genotype in the outcomes. These might become not the dosage, but the predictor markers rather than the actual utility as a dose finding studies. And when you look at 680, as the woman ages, there's a significant difference in the behavior of 680 genotype. So we found women with over 25 with a AA genotype required higher total dosage, suggesting that dose-related modulation of receptors plays a role. So on the impact of the pregnancy rate, again, when you're looking at the lowest outcomes, if you look at the second slide, lowest outcome combination, FSHR 680 A by A, 312 A by A had the lowest clinical pregnancy rate, while another genotype had the lowest live birth rate. This might be useful for somebody who's postponing the pregnancy or counseling. What are the chances with the given number of oocytes? If you get 8 oocytes, your counseling might play a role rather than in the dose of your stimulation protocol. In LHCGR, it plays a major role of LH supplementation, but when it comes to FSHR receptor, this data at the present moment is useful in helping to counsel the patient, or when it comes to oocyte freezing, the number of oocytes required for a particular genotype might vary. That's the carry-home message. So the dose implications is on the left side, you have the 680. On the right side, you have the 29. There is modest but slightly difference in the stimulation protocol of 200 to 350 units across the subgroups. And whereas in a 29, that is not equally seen. So when it comes to FSHR, phenotype is the first and the most important standard. That means your stimulation should be primarily guided by antral follicle count, age, and BMI as the starting dosage. The genotype may help in fine-tuning your dosage. But when it comes to LHCGR, the data is very, very clear. But when it comes to FSHR, you can't use at this present moment with the Delphi consensus and these 2 PhD papers that it will help to counsel the patient of number of oocytes required to get a pregnancy or fine-tuning it, but it's not the primary indicator to plan a stimulation protocol. That's the carry-home message I would like to give.
Thank you, sir. Can we just clarify a few doubts which are there in our minds, sir?
Yes, go ahead.
Sir. So a question here is, patients with immature eggs, only GV, does receptor polymorphism play a role when the genetic testing is normal?
The answer is no. because it depends on the trigger. It depends on the androgens that produce a sudden rush when the LH is when the LH surge occurs with the trigger. That's a completely different mechanism, and its role in the immature oocytes has not been proven in any studies, neither in our study.
Okay, sir. So you were telling us about isolated use of FSH receptor polymorphism should be used in research setting or in patient counseling tool or number of oocytes that might be required when you go for oocyte cryopreservation. The doubt here is sometimes in PCO patients, there can be a combined FSH and LH receptor polymorphism leading to hyporesponse. And at the same time, there can be a variant polymorphism that can cause high risk for ovarian hyperstimulation. So how do we, kind of do polymorphism testing in PCO? Are we really justified in doing it based on the phenotypes we encounter in our day-to-day practice?
In the PCO4 subtypes of phenotypic groups, one is the 4 groups which we see. There, you can find in a PCO LHCGR G by G, 312 G by G, and also LHCGR A by A. That means in some PCO, we also give LH supplementation. So there, the data is very clear and our experience is very clear, and we did not land in hyperstimulation, neither cancel cycles, neither abnormal cohort of abnormal set of oocytes. But you can't use the FSHR data at this moment to define individually that this is the group and this is the dosage. If we look at LHCGR G by G, we said 75 units is adequate. And A by G, we said 37 units is adequate. And AA, no LH is needed. There, it's about preventing a hyporesponder. But when it comes to FSHR, the data is more complex and more subtle, and subgroup analysis and patient subpopulation will determine the future. At present, you can't use FSHR to plan any stimulation protocol. That is our experience. We are only trying to modify and being careful about the follicle to oocyte output ratio in some of these patients. But we have not moved to the quality. So my carry home message is, if somebody is doing an FSHR receptor, unless they do it for every patient and start doing the subgroup analysis, it will not be useful. The same thing can't be said for LH receptor. In LH receptor, we are very, very clear, and that was as far as 5 years back, we could identify that pattern and say, this patient, this is the dosage. The same thing we are not able to say about FSHR receptor, but we are finding patterns which is telling at the oocyte efficiency. If you pull the number of oocytes of the particular genotype and study them, then you get the parameters of these subtypes will respond this way. That is our future next step of study. For example, LHCG, FSHR 680 G by G, 29 G by G, 307 G by G, LHCGR G by G, and 6 base pair normal had a different behavior compared to a 6 base pair LHCGR A by A. So when you're looking at all the combinations, a pattern is evolving. And we are finding totally 64 types of data sets are there. And some of them, when you look at the 1700 patients, the 64 data sets are not equally distributed. That means you're finding the reproductive performance might be different in some of them. So the carry home message is the Delphi consensus, and this work shows in a routine clinical setting, it is not yet fully validated as a clinical tool. But that's not the similar case with LHCGR. Second thing is, we're also finding that it will have a role in low responders to identify in those subgroup analysis, it will help us. Also in patients who are going for oocyte freezing, there till now nobody has determined what is the optimal number of oocytes a patient needs it. Maybe this data will help them in combination with the follicular fluid steroid markers. That's my personal opinion.
Okay, sir. So as you rightly was telling us about advanced maternal age, like above 35, generally we know that the receptor expression is also coming down and the signaling is also different. So most of us in our clinical practice routinely are using HMG in our advanced age group patients directly. Aren't we justified in using recombinant FSH plus HMG in this group of patients where we have a combination of, receptor sensitivity that is happening? We will be better off in stimulation in these group of patients when we use both, sir?
Even the recombinant protein FSH and the FSH in the HMG is not structurally similar. There I personally feel when the molecule gets attached to the FSL to the receptor, each type of protein has a different behavior. And you are producing two types of protein to get attached to the receptor. Then the behavior has not been studied at the granular cell level. And we do not know whether there is a synergistic action or a negative action. We do not know. And that has been never published or studied by any groups. The attachment of the receptor, whether it's recombinant FSH or from the protein that is from the HMG, the urinary protein, when they attach to the receptor, the binding capacity is different. And the dislocation of that is different. So I personally feel not to use two different types of protein in a stimulation protocol. That's my personal opinion.
So given a choice, you would use recombinant FSH plus recombinant LH in a patient who is advanced maternal age, sir?
From 2012, we have been using only recombinant for every patient.
No, I understand, sir. So we just want to understand in advanced maternal age group. So it is a combination of recombinant FSH plus recombinant LH. That's what.
Yeah, suppose if you look at on the second day of the cycle for advanced maternal age, if you do FSH LH, you find a reversal of FSH LH ratio. The FSH might be 5 and the LH might be 2. There you have to give LH supplementation. The HMG HCG has a different type of action when it comes to the luteinization, when it comes to the proliferation, and it has the LH molecule has anti-apoptotic action, and that has a apoptotic action. So when there is a reversal of FSH LH in a PCO, you see 1:2 ratio. In a normal woman, you say FSH LH is 1:1.1. But when the age comes with borderline ovarian reserves, you find a reversal of FSH ratio where FSH can be 6 and LH can be 4. that is, the patient also needs LH supplementation. There, either stick to a single product of HMG without addition of recombinant FSH or LH, or if you are following a recombinant protein, add LH supplementation there. That is my advice.
Okay, sir.
Can I ask a question?
Yes, doctor.
Good evening, sir. This is Vani here.
Yes.
Sir, sir, a couple of questions. You said in this FSH polymorphisms, the requirement of FSH was high. How was it determined? Like number of days of stimulation required was high. That is one question. And another one is in PCO also, I know that you go for long protocols. And there, the FSH is the one which is causing hypostimulation. So LH, you're determining the how much should it be, like 37.5 or no LH or 75 based on the LH receptor polymorphism. You will control your LH, but how do you determine how much of FSH to use? And because you never land in hypostimulation, how do you avoid that? So these two questions for now. And
First thing is, once we finished the study, we started using antagon in some patients, but the criteria to use antagon is AFC 30 and AMH above 4 to 5 is the criteria when we use antagon protocol. There, we also estimate the testosterone and try to reduce the testosterone with metformin before we start the stimulation protocol. And there also we are not using freeze-all protocol, and we're using fresh stimulation protocol. That's when it comes to the second thing is, when we do the starting dosage, we do the starting dosage based on the age, AMH, comorbidities. I'll just share the slide. If you see, my slide is visible.
yes, sir.
Yes, sir.
So this optimal dosage one should determine for their own clinic, and it varies from product to product. And we took optimal dosage of 200 units. And then when you look at the age, when you look at this control, you have a control here. You have a control here. If the age becomes 25, I minus 25 units.
Okay.
If the age becomes 20, I minus 50 units. If the age becomes 33 plus 25, if age becomes 37 plus 50, but we never cross 300 units. Similarly, when it comes to BMI, if the BMI is 22, we minus 25 units. If it's less than 20, we minus 50 units. If the BMI is 30, we add 25 units. If the BMI is 35, 50 units. Similarly, we base on the AFC. If the follicles are between 8 to 16, that's the control. If they're more than 16, we minus 25 units. If it's more than 25, we minus 50 units. Sometimes your FSH with this protocol, and on the right hand side, endometriosis, psoriasis, collagen disorders, thyroid antibodies, they need higher dosage. But on the total, we never cross 300, even though this crosses 300. But sometimes we come as low as 75 units of FSH with this formula. And to this, we add LH polymorphism to add the LH supplementation. Be it antagon cycle or agonistic cycle, we found the results to be similar. This starting dosage, if you're using multiple products, how can you use a same starting dosage? If you're using a HMG and an FSH, you don't know the potency of the product because it's not in MCG, it's in the units, and that's based on the corpus luteum weight of the rats. So you have error. Stick to one product, optimize your dosage, and we have been using this. And when Sandros and Carlo Alvigi were quite interested and surprised that we are using this formula for the last 10 years because they were struggling with the dose finding studies.
Yes, sir. That's the biggest question in the stimulation of
But
particles.
we have been using it for the last 10 years.
I think you've standardized it for your recombinant FSH, sir. All of
Yes.
us have to standardize.
One thing is we use only a single product of everything.
Yes, sir. That's a very
Uh,
important point for all of us.
Like nobody said HMG is inferior. Who said that? But stick to one product so that you know the behavior of that particular molecule. And the criteria for me for saying a hyperstimulation is admission as a criteria to define as hyperstimulation. If the patient comes with complaint of pain and a certain amount of fluid and needs admission, that's the criteria to define hyperstimulation in my protocol. I have to have some criteria, and the criteria are very vague about hyperstimulation.
Okay, sir.
Yes.
Sir,
this
there
has
is a
worked. This has worked very well for us for the last 10 years, and this has also did not raise any premature elevation of progesterone. And the need for stimulation protocol adjusting the dosage occurs in less than 10% of the patients. So when you're not adjusting the dosage, you're not picking up unnecessary for small follicles. You're not recruiting your cohort is uniform. And your wastage of oocytes is less.
What about this Pergoveris? They have a fixed combination and.
No. What happens is, what I'm saying is, all products are good. Stick to one product.
Okay.
Understand your stimulation protocol. Understand the molecule which you're using it, and then use that molecule so that IVF has so many variables, and the greatest tragedy of IVF is monitoring of a stimulation protocol did not change in the last 40 years. Our drugs have changed, our stimulation protocols have changed, but our tools for monitoring a stimulation protocol did not change. No development has occurred. That does not mean there are no tools are not there. Only good quality research did not occur in the area of monitoring a stimulation protocol. What was done at Edwards' time or in the early years of ultrasound and the E2 levels and LH levels, those three parameters, apart from that, nothing has changed. That does not mean no markers are not there.
Okay.
Sir, when your earlier, Luveris was available, so we could add 37.5, etc. to the stimulation. Now, instead of Luveris, are you using Pergoveris? And if you have to use a very low dose recombinant LH, how do you titrate it in Pergoveris, sir?
I suppose I need 225 units of FSH and LH LH 75. There I give 75 units of Bonal F and 150 units of Pergoveris. So that becomes 225 and LH 75. If I need a 37, I use pergoveris 75 units, that is FSH 75 units and LH 37. In fact, the biggest surprise we had was after adding pergoveris, the cost of a cycle has decreased because earlier there was a need for buying the loweries and the FSH. A certain decrease in the cost has come in.
Oh,
A decrease of 8 to 10,000 has come in after we started using. Initially, we were very skeptic that our cost will escalate by 20,000. But what we realized was, but by using pergoveris only for the LH75 and LH37, where you get 150 units pergoveris or 75 units, then that is adequate because they come in vials, multi-dose vials. Then add Gonalef, because you should not use two types of recombinant protein.
okay.
Similarly, you should not use two types of FSH. One is urinary and one is recombinant, because the binding capacity is different for each of the molecule. On top of it, if you overlay the 29 G by G, Kavitha Lakshmi, because in 29 G by G, the promoter region, the amount of receptors that are available is decreased. So the binding capacity is lesser. And on top of it, if you have an inferior product, then you do not know the binding capacity. Kavitha Lakshmi, can you explain the 29 G by G?
Sure.
What exactly happens?
Okay, sir. Sir, actually, this polymorphism is in the promoter regions. Mostly a promoter region, it is present in the upstream of the gene, almost more than 10 nucleotides away from the transcription start codon. And the RNA polymerase, an enzyme which is useful for making mRNA copy from the DNA in the nucleus, it enters through this promoter region and walk along the gene for a while until it reaches the transcription start codon and then starts transcribing the mRNA copy and stop once it reaches the stop codon. This precursor mRNA then it gets modified into mature mRNA and it enters into the cytoplasm where it is recognized by the ribosomes and then FSH protein was synthesis takes place and this receptor gets embedded in the cell membranes. And this promoter region, it is also a binding site for many transcription initiating factors. So any alterations in this promoter box, it may result in the alteration in the amount of the receptor produced. So it doesn't alter the sequence of the nucleotides in the mRNA or amino acid in the protein.
So if the amount of receptor is lesser,
Yes.
and then you have a different molecules, the binding capacity of each, you will be producing completive binding at the site, which is based on the charge of the molecule, the positive charge and the negative charge, the amount of charge that protein has. So a particular protein has a higher opposite charge, then it binds it much more longer. Then the other protein may not have space to bind. That's the actual reality when you look at the receptor binding activity. Can you explain that, Kavitha Lakshmi?
Sir, actually what happens here was instead of a guanine, if adenine was there, it is disrupting the transcription binding sites. So they're not able to, the transcription process is getting affected, sir, here actually. In studies, actually they discovered.
So in that context, by giving two molecules Are we helping or disrupting? I do not know.
Maybe by changing the amount of the dose, just we have to see. I think
No. What I'm saying is, if you're giving a urinary protein and a recombinant protein, the charge of each particular molecule will be different. And that makes the efficiency of its binding, and the internalization sometimes happens with certain receptors. Certain receptors, the receptor gets exhausted and the molecule is re-released. So those are the areas are still evolving, and we don't have much expertise in the chemistry of molecular binding at the receptor. But the carry home message is, I would prefer not to mix molecules because of the receptor binding activity. But I would say that efficiency comes down of the intention we have.
So while we are on that, sometimes we use either clomiphene or letrozole to our exogenous FSH to have an endogenous FSH component as well. So does that combination also works the same way, or is it any different?
Because what happens is, if you look at it, depends on the age of the patient. An younger woman and an older woman has a different glycosylation of the FSH molecule. So those also makes a difference, because the FSH amount of glucose binding sites are different in a younger woman and older woman.
Okay.
That plays a role, but not much studies have been done in relation to the protocols and its efficiency on the receptors, because we are only looking at whether we are able to produce a oocyte in a poor responder, but we are not looking at how efficient that process is and what is happening to the oocyte. Are we producing nuclear maturity and cytoplasmic maturity, which the embryologist should answer? And they should give the feedback about the oocytes, whether while they're doing the ICSI.
Okay.
Whether
Actually, there is a question on the chat box which is there, sir. Does FSHR polymorphism patients present with any particular type of oocyte dysmorphism?
The answer is no. The answer is we could not find anything. But when you look at LH receptor gene, LH gene polymorphism, they're associated with certain dysmorphisms, but none of the receptors are associated with any dysmorphism as per the current literature and as per our experience.
So, do you add LH in the second half of the cycle for final maturation, or continue only with Rec FSH, sir?
No. We suppose if there is a polymorphism, we add LH from day one, be it antagon cycle or be it antagonistic cycle, or even in the recently for one of the random start cycle for a ovarian malignancy patient, we did the receptors quite early, did the progesterone, we gave the progesterone to prevent the luteal prevention, but we added the LH, and we could find the response of LH in that particular polymorphism is not blunted by the oral progesterone. We have a policy of starting it right from the stimulation protocol from day one, be it agonistic or antagonistic protocol.
So you're testing for all patients, sir? That means in your center polymorphisms?
From 2012. We started charging only from 2018 after publication of the article. Till then, we did not charge because we felt it's unethical to do that. after the 2018 publication, we started charging for that service. We do it for every patient, and every patient is based on the LH polymorphism. We start when no LH, LH75 or LH37.
Sir, if you look at different blood groups, sir, so there is some association of O blood groups having lesser response compared to, A blood groups having better response. Is there any link with polymorphisms and blood groups, sir? Not genotypically, at least functionally. Did you find, sir, anything in your research or something?
No, we have not looked at that aspect. That's an interesting aspect. We didn't look at it honestly.
Because there is some patients in this particular group can hyper respond. So I was just thinking maybe it is a different locus genotypically, but functionality wise, will there be any?
I'll ask them to look at the data from that point and then give a feedback, because for all these patients, the groups were there. Any particular pattern is there in different groups.
Okay, sir. Sir, and also your lot of research in FSHR. Have you also done estrogen receptor polymorphism, sir? That also will give us a lot of insights in implantation or anything. It's all research only, sir. Since you are doing in this field, I just thought to ask the alpha and beta receptors.
That's our next project, looking at progesterone resistance and looking at estrogen receptors and progesterone receptors at the endometrium. That's what we want to look at the next project. Continue with this project, but over and above that, we want to add for the stimulation protocol AMH polymorphism is one thing we wanted to add next. And at the endometrium, look at the progesterone resistance and look at the endocrine receptors in the preliminary. At a later point, go into the immunological aspects. Because each is a thread, and if we untangle it, it's becoming a knitting ball, which we are tying around ourselves. And to untangle it is taking 2 to 3 years to understand basic 1 point of clinical practice.
So,
I would say that to just say that this group of patients need LH supplementation took us 8 years to deliver the message. That's a single line in a scientific literature.
So just to understand, since you're doing it since 2010, all of us, for us, when should we actually do LHCGR testing? Just summarize in Poseidon 1 and 2, definitely should we do, or what is your advice to all of us?
For LH receptor, I would say that if you're using recombinant FSH and if you have started the concept of ongoing hyperresponder, that means with adequate FSH dosage and follicles are seen, but the rate of growth of follicles is lesser than desired, or the E2 level rise is lesser, there add LH supplementation. That's the first step one should practice. After practicing that for 6 months, then you know who needs LH supplementation and what's the benefit. Then ask for LH receptor for every patient to improve your results. Without doing that first step, why I said the first step is, the first step will help you to stabilize your starting dosage of your stimulation protocol. Without stabilization of the starting dosage of the stimulation protocol, all these polymorphisms are waste. So I would emphasize that stabilize your starting dosage with the fixed metrics. Then look at ongoing hyperresponder. There add LH supplementation. Then only after using it for 6 months, then move to LH polymorphism as a test for every patient. Regarding FSHR polymorphism, I'm not recommending it because it is too complex at this moment of time.
Sir,
main message here is we should not be using this as a counseling tool for patients who have had a poor response. We should do
that is what is happening. poor response and LH polymorphism, FSH polymorphism have nothing to do with that.
Sir, can anyone replicate your model of starting dose, calculating the starting dose?
Now Sandoz and Carl Alvega started it.
Okay. So this means if we have to calculate LH based on the receptor polymorphism, do you think it's better to use your idea of protocol for fixing the starting dose and then using this?
Because what happens is the whole concept came of adequate stimulation, adequate starting dosage.
Okay.
A hyper-responder is a subgroup of patients with adequate starting dosage. Your follicles do not grow because of LH polymorphism and they need LH supplementation.
Okay.
But without adequate starting dosage, none of them will work.
Okay.
There is a question here. Can we add LH for all patients without checking receptor polymorphism?
No, I never said you should do LH polymorphism. I've said with adequate starting dosage, if you're getting a hyper response and your follicles are there in the ultrasound, but they're not growing, provided you give an adequate dose of FSH, then add LH. LH is not needed for every patient. So the question of adding for every patient does not arise. These are subgroup of patients. In fact, in 2019, the ICMAT has added hyper responder as a third group to poor responder, hyper responder, and hyper responder. That means the concept is evolving only in the last 5 years.
Okay.
So for a hyper responder, the whole thing is with adequate FSH dosage is the first thing.
So can HMG also be used in this way?
Yes. In fact, what happens is, provided the quality of product is consistent, I would also advise dilute 1 ampule of HMG and estimate the HCG in that. If there is too much of HCG, don't use that batch. Use a different batch. That way you can decrease the amount of HCG in the HMG. Some batches will have 75 units, some batches of HCG, some batches will have 150 units, and the bioequivalent of HCG and LH is vastly different. But you can dilute a vial and estimate the HCG, and different batches will have different HCG, and use the batch with the lowest HCG. That way, you're avoiding the premature progesterone rise and also a much lower LH supplementation, where it is actually the HCG you are supplementing it.
Yes.
That is one way of, we used to do that between 2005 and 2010.
So the same model to fix the starting dose of HMG.
Yes.
Okay.
Thank you so much, sir. You wanted to say something, sir? Please.
No, I'm not sure whether we did justice, but we wanted to throw a light.
No, sir, this is such a complex topic, and with presentations from PhD scholars who have done research in these polymorphisms, and with your practical insights, at least we could understand what these polymorphisms are and why we should be using it, how to standardize the protocols, and why not to mix two different brands and two different molecules of different action. Lot of practical insights we have, sir. We should understand the evolving science also, and no other expert other than you who has done so much research since 2008 could, tell us in a 1 hour session. So that was really a cooked recipe for all of us. Thank you so much, sir. And thanks everyone for interacting and asking your questions with sir. Thank you so much, sir, for spending, and thank you, Dr. Kavitha and Dr. Kavitha Lakshmi, for such a wonderful presentation and your insights on your PhD thesis. It was really wonderful to host all of you in this masterclass. All of us benefited a lot from this session, sir. Thank you so much.
Also, one question to sir,
Yes,
if
ma'am.
I'm allowed, Dr.
Yes,
Kundavi
ma'am.
here.
Please.
Yes.
Sir, like in a hyper responders, the mega sets trial, and everybody said you start an highly purified HMG so that you can avoid hyperstimulation. Whereas we use the same HMG in poor responders and hyper responders too. So how does it differ, sir, in hyper, hypo, and poor, the role of HMG?
Honestly, I stopped using HMG since 2012. So my experience with HMG is very less.
Okay.
I've been using it, using recombinant because they discovered pyrons or prions in the lyophilized powder of HMG. And that's the reason Australia has banned the use of HMG for safety reasons. So you don't get HMG in Australia. And the HMG that is available in US has a different manufacturing process compared to the one that's in India. So we are looking at the US data, which shows that using HMG has the FDA regulation of manufacturing it is slightly different than what the world over follows that. So, regarding your question, since I don't use HMG, I won't be able to comment on that.
Okay, sir.
Because I feel there's too much of batch-to-batch inconsistency in that product.
Even with the highly purified, sir, like Menopure and the Menatos?
What happens is we also find 5 to 6% batch-to-batch variability in recombinant protein. So even though the data shows there's no batch-to-batch variability, we are finding 5 to 6% difference in batch-to-batch variability in recombinant protein. Because we make it a point to write which batch we have used in a particular patient. If there's a change in batch, we also document that. And we are finding a very small difference in the recombinant protein. So what we do is, for each file, we write the batch that is used, and you might be able to pick up the difference because once you start analyzing the results of the total ooze follicle to oocyte output based on the batch number, you get certain insights. That's my suggestion.
Thank you, sir.
Thank you, sir.
Good.
Thank you, sir.
Thank you, everyone. Thank you so much, sir,
Thanks.
for this session.